Good morning, and welcome to our Alaska Collaborative on Health and the Environment teleconference on environmental contributors to breast cancer, the underlying science and opportunities for action. My name is Diana DeFazio, and on behalf of Alaska Community Action on Toxics, I'll be facilitating today's call. And for those of you who aren't familiar with CHE Alaska is a regional partnership group of the National Collaborative on Health and the Environment. CHE Alaska aims to advance knowledge and effective action to address growing concerns about the links between human health and environmental factors. You can find more information at akaction.org and at the national website healthandenvironment.org. Today, we are truly delighted to have Dr. Meg Schwarzman as our presenter. Dr. Schwarzman will be traveling to Alaska next week to meet with medical professionals and to give a public talk in Anchorage, and we'll tell you more about those events at the end of the call. Everyone who signed up for the call as of yesterday received a link to the presentation slides, which you'll need to download to follow along. And if you downloaded the document yesterday, please note that it has been updated. So we'd appreciate it if you could go back and follow the link again. If you signed up more recently for the call, go to our website, akaction.org, scroll down to the CHE Alaska call post, and you will see a link to the PDF there. And as with all of our CHE Alaska teleconferences, we will leave time at the end for discussion and questions from participants. So now it is my pleasure to welcome and introduce our featured speaker. Dr. Meg Schwarzman is Associate Director of Health and Environment for the interdisciplinary Berkeley Center for Green Chemistry. She is an environmental health scientist in UC Berkeley School of Public Health, where her research and teaching focuses on endocrine disrupting chemicals, U.S. and European chemicals policy, and strategies for applying environmental health knowledge to the design and selection of safer chemicals and materials. Dr. Schwarzman also practices medicine part-time. She has a medical degree from the University of Massachusetts Medical School and a master's in environmental health sciences at UC Berkeley School of Public Health. And welcome, Meg. You may go ahead and begin.
Thanks so much. I think I've unmuted my line.
You have.
I really appreciate your having me on the phone and, of course, having me up to Alaska next week. I'm excited to be there and looking forward to our meetings and presentations then. As Diana mentioned, I'm a physician first and environmental health scientist second, and on this topic, those two pieces of my training come together well. I'm interested in the role of the environment in breast cancer for two main reasons. One is the sheer prevalence of breast cancer. As most people know, I think it's the most common invasive cancer in U.S. women, and the current estimates are that 1 in 8 women will be diagnosed with breast cancer in her lifetime. So this affects many of us, either personally or through family members. And it's also, although treatment and diagnosis has gotten better over the years, it's still the second leading cause of death due to cancer after only lung cancer in the U.S. And the second reason is that there's this emerging evidence for a connection between chemicals in the environment or exposure to chemicals in the environment and breast cancer risk. And although that can be scary and disheartening, for me it also translates into a really profound opportunity for prevention. So while I'll give some alarming evidence today, I also want to end with some things that we can do and ways that we can work both individually and collectively to reduce our exposure to chemicals in the environment and ultimately, hopefully, to reduce breast cancer risk. So first I'm going to go over sort of these clues that we've gotten that the environmental exposures raise risk of breast cancer. So if you'll go to the next slide. There is at least a 10-fold variation around the world of breast cancer incidence rates, with higher rates in more industrialized countries. So you see on this map, although it's small, the darker shaded areas are the areas with higher incidence of breast cancer, and this is back in 2012, but it looks similar now. And although that would be striking on its own, what's really particularly striking is that when people migrate from areas of low incidence to areas of higher incidence, their own risk of developing breast cancer changes. It rises, particularly if they migrated as children. And more strikingly, successive generations, that is, the children of migrants, have a risk of breast cancer that more closely resembles the rates where they live, that is, where their parents move, than their parents' rate of breast cancer. So this hints that there is something, there's a a more, perhaps more influence of location than of genetics, or at least a significant influence of location on risk of breast cancer. So we also see that in the areas with historically low incidence of breast cancer, those are often changing and increasing as lifestyles westernize, and that sort of westernization of lifestyles is a bit of an ill-defined surrogate for changes in other factors such as childbearing age and number, dietary habits, and then it's also posited that it changes in exposure to carcinogens and exogenous estrogens, that is, estrogens that come from outside the body rather than what's natively expressed in the body. And so those breast cancer, as areas that have not typically had very high rates of breast cancer, as they sort of westernize and industrialize, the breast cancer rates in women in those areas starts to resemble the rates for women in industrialized countries. This is sort of provocative in and of itself. And if you go to the next slide, the other element is that genetics appear to explain anywhere from 5 to 27% of breast cancers. That 27 number makes it look like we have counted this really, that researchers have determined this more precisely than it has been determined. But we know that inherited gene mutations, so like this is like BRCA1 and BRCA2, which folks have heard of as being sort of the breast cancer genes, are a small portion of this. And then also there's inheritance of physical traits that raise a person's risk of breast cancer, such as breast density. And this evidence comes from some really interesting studies that were done in Scandinavia where they looked at identical and non-identical, so fraternal twins, and they were able to evaluate so they looked at these identical and fraternal twins who developed breast cancer, and they by comparing the rates in those two groups, they were able to evaluate the proportion of breast cancers that were attributable to shared genes, and the proportion attributable to shared environment, and the proportion attributable to non-shared environment. So that would be like the genes is clear, but shared environment is like if one of their parents was a smoker, or if they were equally exposed to some pollutant or other chemical carcinogen. And then a non-shared environment would be like if one of them was a smoker or heavy alcohol use and the other one wasn't. So I find those very intriguing studies because they're able to look at these pairs of people who are different degrees of genetically with shared genetics and also with shared environment, and start to tease that out. So if only 5% to a third of breast cancers are explained by genetics, that really leaves the majority to these other factors: the environment, potentially chemicals in food, air, water, consumer products, and workplaces, and I'll go into those in a little more detail. And also these reproductive factors that are not genetic, but some of them may be a little bit linked to genetics, like age at puberty and age at menopause, but also childbearing, delayed childbearing, not having very many children, which raises the risk of breast cancer slightly, and also whether you breastfeed. And finally, lifestyle choices, such as how physically active you are, whether you smoke, and how much alcohol you use. And I just want to note that, until recently, I think, the popular press and sort of our popular understanding really emphasizes these lifestyle factors. And the thing that I don't like about that, although they are relevant and they are under our control, so they're potentially places of intervention, it also puts this onus on women. And what I want to go into today is what these other factors may be that are less under our control and that we as a society really, I think, need to be doing something more about. So I want to look at these breast cancer risk factors in a little more detail because of the established breast cancer risk factors, a majority of them actually appear to have this mechanism that we can reduce to some hormonal factor or to exposure to a carcinogen. So the next slide summarizes some of these known breast cancer risk factors. And I just want to acknowledge some of these slides are not exact copies of, but based on some ways that the Silent Spring Institute has presented this information. Those slides are noted on the lower part, but I wanted to just acknowledge Silent Spring Institute that does a tremendous amount of excellent work in environmental contributors to breast cancer and a lot of education around it. So these known breast cancer risk factors include a couple of genetic factors, but also exposure to things like ionizing radiation, combined hormonal replacement therapy, tobacco, smoke, and alcohol. We've talked about alcohol, overweight, particularly after menopause, lack of physical exercise, also these reproductive history factors, and just recently identified by the International Agency for Research on Cancer is shift work that disrupts the circadian rhythm. And that shaded area points to this majority of breast cancer risk factors that reduce to exposure to a carcinogen or a hormonal factor, even if it's not external. So these reproductive history factors are actually, they all kind of reduce to lifetime exposure to estrogen. And the larger your lifetime exposure, a woman's lifetime exposure to estrogen is, as a result of, for example, early menarche or late menopause, the higher that exposure is, the more likely she is to develop breast cancer. And so this is part of what hints at hormonal and carcinogenic chemical exposures, whether they come from the outside or the inside. I want to talk about each of these in a little bit more detail, because the good news is that anything but inherited risk factors are at least theoretically preventable. So if we understand those a little bit better, we might be able to start taking steps to prevent them. So the next slide goes into a little more detail on the impact of combined hormonal replacement therapy. And I realize I think I wrote just hormonal replacement therapy, but specifically these studies are looking at hormonal replacement therapy that is both estrogen and progesterone combined, because that's not the only kind of, I'll call it HRT, hormone replacement therapy, that is used. And there's an interesting story here, but as context, I want to just note that breast cancer isn't a single disease. It's really a diverse set of diseases, and each has a distinct cause, and also it has a distinct pattern of progression. And of course, because of that, the treatment depends on the features of the breast cancer that you have. So part of that kind of muddies the picture, but it also helps us start to distinguish among what the mechanisms might be that are contributing to breast cancer. So I think a lot of people have heard of the Women's Health Initiative that was done in the 90s and early 2000s, and it was actually ended early because of, or some results were released early because of findings in 2002 that showed increased breast cancer rates among women on combined hormonal contraception. And this graph shows breast cancer rates in the U.S. across different years, and that arrow points to 2002, at which point breast cancer rates in older women start to decrease demonstrably. And you can see that the lowest line on this, the bright green, is women under 45. And although these rates appear to drop off similarly in 2002, statistically that line over time is actually flat because there are these fluctuations over time, but they're not necessarily statistically significant changes. But for women over 45, that is postmenopausal women who would, or more likely postmenopausal, who would be more likely to be taking hormone replacement therapy, the incidence of breast cancer in that age group dropped dramatically after the release of these data that tended to mean that women either stopped taking HRT or never started taking HRT. So when the results were released showing that the risk of combined hormonal replacement therapy outweighed the benefits, many women stopped taking it or never started. Then there's been some interesting sub-analyses that have found that this drop is mainly driven by reductions in ER-positive tumors, so that's estrogen receptor-positive tumors. This gets at the point that all breast cancer tumors are not created equal, and they express different receptors. Some of them are sensitive to estrogen, some to progesterone, some to both, and some to HER2, which is a human epithelial growth factor. But in any case, we don't have to go into that, but just to say that there are multiple versions of breast cancer, and it was particularly these estrogen receptor positive tumors that dropped off. And we also saw that primarily in white women from higher socioeconomic status counties. And that supports the hypothesis that this rapid decline in breast cancer cases may be attributable to decreasing HRT use because they're ER positive, so that means that they're going to respond to estrogen and respond to the lack of extra estrogen that's included in HRT. And also this is a demographic that tended to be higher users of combined hormonal replacement therapy. So the final point in this is pretty significant in that the release of these results, and also to some extent an earlier study that had started to raise flags about HRT risks, and that was in the late 90s, and I think that's part of why you see the risks start to drop a little earlier than 2002. But there's been a study, follow-up studies from the Women's Health Initiative that looked at the impact of this decrease in HRT and subsequent breast cancers. So if you look at the women, or at the period between 2002 and 2012, they estimated that as a result of these studies, there would be over 4 million fewer users of HRT, and as a result, as many as 126,000 breast cancers avoided, and 76,000 cases of cardiovascular disease, so that's like heart attacks and strokes, which were also increased because of hormone replacement therapy. The drop in HRT use also is attributed to about 260,000 more fractures. But since those are so much less serious than either cardiovascular disease or breast cancer, what they saw was a total of about 145,000 more quality-adjusted life years. So that's just to say women were living longer and higher quality lives as a result of not using combined HRT. And I think I wrote this number slightly wrong. It saved $35 billion in expenditures, not 34, which I think is on the slide. And so they looked at what is the return on investment, basically, of the study, and it was over that. It was about $37 billion of these direct healthcare costs, but also other associated costs. So that's a pretty striking public health intervention. And it hints that if other exposures, if other chemicals work like hormones, they may also be contributing to breast cancer, even if not this dramatically, and there's potentially something we can do about it. So if you go to the next slide, there's this question of, could chemicals in the environment act similarly to the hormone replacement therapy that we saw as playing a role in breast cancer risk? We see chemicals with similar properties, both direct carcinogens, things that damage DNA, and also chemicals with hormonal activity that may pose risks of breast cancer. So these are some examples of chemicals that are linked to breast cancer or possibly linked to breast cancer, where the evidence isn't 100% clear. And these chemicals all have different mechanisms. So benzene and 1,3-butadiene, which are released in gasoline vapors and also in vehicle exhaust, are probably mainly direct carcinogens in that they damage DNA, but perfluorinated compounds like PFOA and PFOS that are used in nonstick coatings and stain repellent treatments of fabric and upholstery are probably endocrine disruptors and act like synthetic hormones. And there's actually some studies looking at Inuit women in Greenland, and they've been found to have much higher risk of breast cancer with as they have higher levels of perfluorinated compounds, which tends to occur in native populations because they're eating higher on the food chain. They're basically consuming food like marine mammals and fish like tuna that live high on the food chain and that have ingested many more chemicals, synthetic chemicals, and they concentrate in their bodies because they accumulate through food webs and this is particularly true of chemicals that persist in the environment and bioaccumulate, that is, build up through food webs. So you can read the others in here, halogenated flame retardants that have been very prevalent in furniture foam and are slightly less prevalent now thanks to some changes in regulations in California, but also still are used in electronics and in several settings like hospitals and airplanes and cars where they haven't been eliminated. Organic solvents like those used in paint strippers and dry cleaning, spot cleaners also, PAHs, polycyclic aromatic hydrocarbons that are in vehicle exhaust of all kinds, and also the chemicals that you eat in charred meat. And then there's also this category of chemicals that are probably contributing to breast cancer basically following developmental exposure, and I'm going to talk about that in a lot more detail. So that's just to say that some of these are sort of classical carcinogens in that they damage DNA, some of them act through hormonal mechanisms, and some do both. So I'm going to look at each of these mechanisms in a little bit more detail as context for understanding why chemicals might act this way. So there's basically these three, to be a little bit simplistic about it, paths to breast cancer. One is direct DNA damage, one is promotion of tumor growth through hormonal mechanisms, and the final is disrupting mammary gland development. So, sorry, that's on the next slide. I didn't say that. And I'll go into each of these in some detail. So if you go to the next slide, there should be a largish picture of, or depiction of DNA, the double helix. And what can happen is errors in DNA. So either through spontaneous damage to DNA that happens as it replicates, so it duplicates itself many times as cells grow and develop, and there can be just spontaneous errors in that, or DNA can be exposed to carcinogens like through ionizing radiation or chemical carcinogens like benzene or PAHs, and those can break the DNA, or they can, like in the bottom of this, there's that little red burst, and that's what we call a DNA adduct, so a harmful chemical gets added onto the DNA. And it doesn't matter, just know that there's a variety of ways that DNA can be damaged, and that damaged DNA can then, we call those mutations, and it replicates these mutated copies of the DNA that don't work right. It usually takes more than one mutation to cause cancer, so there can be one mutation and then a subsequent mutation and a subsequent mutation, and ultimately you get to a cancerous cell. There's also, our body has DNA repair mechanisms. So sometimes there will be a mutation and the body repairs it and it goes on to produce normal cells. That's certainly the case in, we're exposed to UV radiation from the sun every day, but we don't all develop skin cancer every day, and that's because of repair mechanisms. So DNA is damaged by UV radiation, or it can be, and then the body repairs those breaks or adducts, and we go on living our normal lives. So if multiple mutations happen and the body doesn't repair it, then mutated cells can persist and replicate rapidly without the usual checks and balances. That is, often normal cells with some DNA damage will die off, but often tumor cells do not, and then they reproduce rapidly. So on the next cell is this second mechanism in which hormones can promote tumor cell growth. This is again a simplified diagram, but to get the point across, estrogens often stimulate cells that have estrogen receptors, like many breast cells. And so the estrogen causes them to grow in normal ways, but if there is DNA damage, if there's a mistake in how it replicates, or if there's mutation from an external carcinogen, then that damaged copy of the cell replicates rapidly. And those cells are still responsive to estrogen. So in the presence of estrogen, either our own estrogen or estrogen mimicking chemicals or estrogen like in hormone replacement therapy, then they would cause those cells to grow rapidly. On the next slide is the third sort of path to breast cancer, which is a little less direct. This is the path in which mammary gland, so we use that term to describe breast tissue in animals because this evidence is from animals, where animal mammary glands are observed, their development has been observed to be disrupted by early, like prenatal or peripubertal exposure to hormone mimics or hormone disruptors. So these pictures are whole mounts, that is to say they took breast tissue from an animal and put it on a slide and stained it and looked at it through a microscope, and they're the mammary glands of rat pups that were exposed either to the dioxin TCDD, which is in the bottom row, or a control substance in the top row, and they were exposed during prenatal development, so they were exposed in utero. And when you compare that normal mammary gland development stages illustrated across the top, and those, the PND is just postnatal day, so that's going through development. The rats exposed prenatally to TCDD, the dioxin, showed early and permanent delays in the mammary gland development. So what you're seeing here in the pictures is basically there's stunted progression of the glands as they grow and spread through the surrounding tissue. There's reduced branching of the ducts, and there's delayed maturation. The ducts go through this development process where they form these terminal end buds, and those form lobules. In any case, it's the maturation process, the growing up process of the duct cells of the normal breast development, and it's sort of like they're arrested. And that's significant because that arrested development corresponds to immature and what we say is less differentiated mammary tissue. And in general, anything, tissue and cells that are less mature are more susceptible to corruption by external influences. We would say the same of children developing, right? Before they're grown up and mature, you can have more influence on them, and they don't have the internal development to sort of stay the course and stick to their own path. I'm joking, but it's just a parallel with sort of undifferentiated, immature tissues, as they're more susceptible to these external influences, such as exposure to carcinogens. So what this illustrates, and we've seen this for more than dioxins, this has been illustrated for many hormone disrupting chemicals, endocrine disruptors. And this highlights the notion of a two-hit model of carcinogenesis. That is to say, early life exposures can alter the growth and development of breast tissue, and that first hit kind of primes the tissue and makes it more susceptible to developing cancer following a second hit, either from our own native estrogen or exposure to a carcinogenic chemical or radiation. That's one theory, and it's important to note that this two-hit hypothesis isn't the only way that endocrine disruptors can promote breast cancer. We've also seen some animal evidence that of spontaneous development of tumors following endocrine disrupting chemical exposure that is without a second hit. And so it's just good to keep those two models in mind. So let's look at some of the implications of this alteration of breast development in people. If you go to the next slide, this is just telling the story of DES, which many people have heard of. It's a synthetic estrogen that was prescribed for pregnant women in the mid-20th century to prevent miscarriage and other pregnancy complications. But there were studies in the 1950s that showed, or late 1950s, that started to show that it wasn't effective. So doctors stopped prescribing it, women started stopped taking it as much, and then there was a cluster noted of adolescent girls with what was a previously very rare form of vaginal cancer, but was showing up in all these adolescent girls, and doctors traced it to their mother's use of DES during pregnancy. And so that brought about the end of DES use in the United States. But it was actually 6 decades before we really understood that there's also a link to breast cancer. And that link is true in women who took DES. There's a slightly increased risk of breast cancer, but it's a little more obvious in the children of women who took DES during their pregnancy. And it was this 2011 paper in the New England Journal of Medicine that really showed there were conflicting findings prior to that about whether it was linked to breast cancer. And this paper really showed it, and the way that it showed it, in this graph at the bottom you can see the red line says exposed VEC, and that's vaginal epithelial changes. And the importance of that is it's simply a marker for exposure to very high doses of DES. So this was a way of distinguishing the women who had been exposed to very high levels of DES in utero prenatally from those who might have been exposed but to less. And you can see that for women who were unexposed or exposed but to not very much DES, they have similar rates of breast cancer, whereas those who were more highly exposed, as indicated by vaginal epithelial changes, they have a higher risk of breast cancer. So there's a few reasons for this. Partly there's disease latency. That is, they were exposed prenatally and they didn't develop disease until they were adults. And it's partly because these links are hard to detect. And if you measure a woman's level of DES now, it's not going to tell you anything about the actually influential stage when she was exposed. And it doesn't correlate with disease rates now. And even in pharmaceuticals where the FDA requires proof of both efficacy and safety, we won't detect all the outcomes. So if women don't develop breast cancer for decades, we're not going to, in those initial stages of safety and efficacy testing, detect that risk. And that's especially true then with industrial chemicals, not pharmaceuticals, for which there's no standard pre-market safety testing required. There's some similar findings with DDT that I won't go into in detail now just for time, but this underscores the point of how important timing is in determining the outcomes following chemical exposure. So if you go to the next slide, there's a fairly complicated graphic that I'll just explain in some detail, superficially. But so we already understand that there are different health effects that result from exposure at different life stages, not even to do with hormonal exposures. Like we know this about lead. So prenatal lead exposure can lead to profound mental retardation, whereas adult lead exposure may, in very high levels, create high blood pressure and some other problems. But the implications are very different. If a developing baby is exposed in utero, they can have much more profound effects. So we already know about this. But it also seems that this is true for developmental exposure to endocrine disruptors. For example, the slides that we saw earlier of the dioxin, the TCDD exposure, what was influential was when that exposure occurred on a specific gestational day in the rats, not before, not after. And so there's obviously these distinct windows of vulnerability. And what this diagram is showing is basically what happens at each level of normal mammary gland development in the rat, and it corresponds to what happens in the human, but we don't have the pictures of those, and how those are altered with exposure at each stage of development. There's these three distinct stages of susceptibility: the prenatal period, puberty, and pregnancy. And that is really, it's almost unique to the breast. All organs develop prenatally, but the breast really is one of the few organs in the body that has these really distinct periods of development and therefore susceptibility much later in life. So there's a very distinct time of rapid growth and development in puberty, and similarly around pregnancy and lactation. And so those are periods throughout a woman's life in which that breast tissue stays or becomes again very susceptible to damage from carcinogen exposure. If you go to the next slide, I'm going to move on to things that you can do about this, because it's kind of alarming. So the next slide is about things that people can do on an individual basis that might help reduce exposure to some known breast carcinogens. So these stem obviously from the chemicals that I listed earlier. One is limit your exposure to gas fumes and exhaust. This includes not just from cars, which can be difficult, but also like gas-powered lawnmowers or weed whackers. Use a rake. So also you can choose furniture and carpets without stain-resistant treatments, and certainly don't apply your own. There's spray-on stain-resistant treatments, and your exposure could be high. It seems hard to imagine how you could be exposed to those stain-resistant treatments when they're on fabric or carpet, but in truth, they don't stay on fabric like couch fabric, upholstery fabric, or carpet. They tend to migrate off, and they tend to accumulate in house dust. And without knowing it, we are breathing, eating, and touching house dust all the time, and we're exposed through those routes. You can try to buy furniture without chemical flame retardants or replace the polyurethane foam in couch cushions. Now, since 2014, there's been flame retardant-free polyurethane foam available, and you can find it by looking at the label on the back of the couch. And if it says TB117 2013, then it conforms with that later standard. If it doesn't have the 2013, it is more likely to contain chemical flame retardants. If it says 2013, it's not 100% guarantee that it doesn't contain chemical flame retardants, but it means it confers with this with the later standard that doesn't absolutely require those chemical flame retardants. This is something that most people don't know. For kids, use cotton rather than fleece PJs because the polyester fleece is required to be flame retardant because it's so flammable. And so often there are added flame retardants. Those are often halogenated flame retardants, and it's been shown that the flame retardants get into the kids because they measure them in their urine after a night sleeping in the PJs. So that's a pretty quick and easy fix. And finally, instead of doing dry cleaning, look for wet cleaning processes, and those are done with fewer organic solvents. Whether that's a significant source of exposure to people who pick up dry cleaning is unclear. It's certainly a significant source of exposure for the people who work in dry cleaning and are exposed to these solvents all the time. And similarly, like one trip to the nail salon may not dramatically affect your risk of breast cancer, but for the women who work there all the time, it may well increase their risk. And actually, similarly for apartments located over dry cleaners, it's been shown high levels of the solvents in the indoor air. So sometimes we talk about interventions not so much to decrease the consumer's risk, but to decrease the risk of workers or surrounding environment. And that's true if you go to the next slide of some of these ways to reduce synthetic chemical or pollutant exposures in general that may not be specific to breast cancer. But these include like eating low on the food chain and also as much organically grown food as possible. And while this may reduce our individual exposure to pesticide residues on food, and it has been shown to, whether that's significant enough to reduce risk of breast cancer isn't known, and other diseases it's not known. But it certainly reduces occupational exposures to pesticides and to exposures of the surrounding communities, which often involve the families of farm workers. And so if for no other reason, it's a worthwhile endeavor as much as possible. Also avoiding products with fragrances. Perfume has a very high level of phthalates. Also air fresheners, scented personal care products, and household cleaners tend to have high levels of phthalates. And again, this goes for the users of those, probably to a higher degree, but even to a higher degree, professional janitors, for example, exposed to scented cleaning products are going to be exposed to higher quantities. Avoiding antimicrobial soaps is an easy one. Hot water and soap works just as well, and you can avoid exposure to the known endocrine disruptors, triclosan and triclocarban. That way, similarly for toys and clothing advertised as antimicrobial, I saw a stapler advertised as antimicrobial. And we don't know for sure what those contain all the time. They may be nanosilver, but we don't know much about the health effects of nanosilver. And they may be these endocrine disrupting compounds, triclosan and triclocarban. You can choose food containers that are not canned foods because the lining of cans contains bisphenol A, and that's released into the food. And again, although it's been shown that eating a lot of canned foods and soups increases your exposure to bisphenol A, the other thing that we know is that workers in factories that generate the resins are very highly exposed to bisphenol A. And even though we don't fully understand all the health effects, it's unfair to those workers to be exposed to what we do know is a very endocrine active substance. Other basic practices about your house: leave shoes outside because that avoids tracking chemicals inside that are in dust. Clean, use a vacuum with a HEPA filter, and clean with a damp rag instead of like a duster or a dry mop. And then washing hands frequently, especially before meals, can reduce ingestion of chemicals such as lead and flame retardants and bisphenol A. So I like to give these lists because it gives people something to do, but there's a problem with this approach. And if you go to the next slide, that is basically that synthetic chemicals are ubiquitous and they undergo long-range transport, some of them persist and bioaccumulate, and this gets back to the point about there being particularly high concentrations in polar regions of the world, and therefore among the communities that live there, who are not generally the producers and primary producers or users of these compounds. Many are in consumer products, as we've already talked about, and there's some examples on this slide. And we get this false sense of security that if it's sold, it must be safe. But in fact, there's very little pre-market testing required of chemicals, and we're still playing catch-up of the tens of thousands of chemicals that are in commercial use. We have data, safety data, real health data on very few of them. The illustrations here are just to describe some of the sheer volume that we're talking about. These tanker trucks are here to illustrate what 72 billion pounds a day looks like. That is the daily production and importation of synthetic chemicals in the US. And if you converted them to equivalent gallons of water, which isn't exact, but if you did, and you filled up tanker trucks with them, 72 billion pounds would fill up enough tanker trucks to line up end to end and stretch 10,500 miles. So it's a huge volume mass of chemicals that are produced or used in the US every single day. And this graph just shows the results of an analysis that we did comparing projected global chemical production in the blue bars to projected global population. I meant to say chemical production, I hope I said that, to global population growth. And we all think of global population as expanding out of control, and projected growth in chemical production is actually expected to dwarf that. And all of these chemicals are new to human biology. Human evolution, is long compared to modern chemical production. And so the notion that, we've grown up with these chemicals and we know how to process them, although we do metabolize the chemicals, there's no sense that we have co-evolved with these chemicals and know how to make them harmless in our bodies. So this just raises the point that individual action is limited in what it can accomplish. And specifically about breast cancer, I like to think about the risk factors in a couple categories. There's, the ones that women can't change. We can't change that we get older, increasing our risk of breast cancer. We can't change our sex, which is the biggest risk factor for breast cancer, right, being female. We can't change when we get puberty or menopause. And there's others that are minimally under our control, the reproductive factors that we talked about. Exposure to secondhand cigarette smoke is often not under our control. And then there's some that we can partially influence that I talked about: smoking, exercise, alcohol use, and some chemical exposures in the ways that I've just discussed. But I'd actually put a lot of chemical exposures into this category of factors that we really can't control for the reasons that I've just described. And we have proof of this in biomonitoring that has found hundreds of synthetic chemicals and pollutants in people, including in umbilical cord blood. So babies who haven't even been exposed to the outside world yet, except through their moms, come out pre-polluted. And so, there's also only so much you can control about your workplace and your home, the environments you live in, where your kids are. For example, it's almost impossible to eat out without consuming foods grown with pesticides or stored in cans. Cash register receipts may be a significant source of BPA exposure or related chemicals, and it's really hard to go through the world not touching cash register receipts, especially if you work in a store. Schools and daycares are often primary sources of exposure to flame retardants, PCBs, pesticides, and some workplaces, as I've mentioned, are particularly chemical intensive, and it's not just factory work, it's also painters and house cleaners and auto mechanics and dry cleaners and nail salons and construction and even nursing. Actually, there has been shown higher cancer rates among nurses who work with chemotherapeutic agents. You can't get in a car or an airplane without flame retardants and plasticizers. And so I say that not to be desperation-inducing, but to be energizing about the need for collective action. And if you go to the last slide, I have some of the ways that we need to be acting collectively to try to reduce our individual and our society-wide and population-wide exposure to synthetic chemicals and pollutants, including breast carcinogens, but also other chemicals. And so I have just a few ways here. There are more to do collective action, and I've given examples of some NGOs and other organizations working in each of these specific areas, because I think they're really interesting ones to get involved with. They're not the only ways, but I just wanted to give you further places to look. You can advocate for better chemical testing and chemical policy reform. It's a year ago today that the Lautenberg Safe Chemical Act was signed into law, which revised our antiquated Toxic Substances Control Act in this country, and it's an improvement. It's not perfect, and there's a lot that needs to be done with advocating for strong implementation of the law. I've talked a tiny bit about some environmental justice issues, but I actually want to say a little more about that in this context, because not only are there persistent disparities in breast cancer diagnosis and treatment, as I think is pretty widely known, there are also really profound disparities in exposure to synthetic chemicals and pollution. And communities of color often bear a disproportionate burden of exposure to multiple sources of pollution. For example, they may be living near freeways or ports, and simultaneously near polluting industries or waste disposal sites, or in housing that has very high levels of lead paint. And we know now that there's a synergistic effect of chemical exposure combined with economic, life, and social stress. So that stress, just being born into poverty, appears to amplify the impact of any subsequent chemical exposures that you may have. And so this is really, I think, in terms of how to have the biggest impact for our interventions, looking at disproportionately exposed communities and areas, regions, is important to think about reducing the risks from chemical exposure. And it's also very relevant in thinking about occupational exposures, which is why I have a separate line here for supporting union health and safety organizing. You know, with the decline in unionization in the U.S., there's a great strain on the worker health components of unions, committees of unions that look out for worker health. And there's also just a growing number of workplaces that aren't unionized. Just as a side note, we saw with the recent very influential revision to the process safety management regulations, that is just to say the industrial safety regulations on refineries in California, that process was driven by a union. It was driven by the steelworkers, the United Steelworkers. And they were really the political and technical muscle behind that intervention. And we want to do the same for the chemical industry in California. There's 1,900 chemical plants that are operating under outdated safety standards, but those plants aren't unionized. And so there's very little mechanism for actually extending these safety regulations to chemical plants. And you know, there's hundreds of thousands of both workers and residents in surrounding areas at risk if these chemical plants blow up. So I think it's important to understand the relevance of unionized workplaces and their health and safety capacity for preventing occupational exposures and exposures to surrounding communities. And likewise, there are some leading companies that are doing, taking a very active role in replacing hazardous substances in their products and their operations with safer substitutes. And some of them are working together, and they're working together not just on safer substitutes, but on changing chemicals policy. So I will end there. Thank you for your attention and patience. I know that some of that went by pretty quickly, but we have time for questions and discussion. I welcome both. I don't have to answer questions. I'd love to just hear your thoughts also. And I'll end there.
Thank you. Thank you, Meg. I want to make sure everyone can hear me. Can you hear me, Meg?
Yes.
Okay, great.
Yes, sorry, I pressed mute briefly.
I got a little, just wasn't sure if I had unmuted myself. Yes, thank you for your wonderful presentation. And we do have time now for questions and discussion. And what you would want to do to participate is press *2 to unmute your line, any of you callers out there. Please also state your name and affiliation. And we'd like to get to a couple of questions, so please be relatively brief. So it's now open to our participants for discussion and questions. Again, *2 to unmute your line.
Hello, can you hear me?
Yes.
I'm interested in what you know about the ingredients in Tetra Paks, because you mentioned that in your slide set.
Yes.
Why did you caution about Tetra Paks?
Can you just tell me who you are?
Oh, sure. My name is Dr. Lynn Patrick. I'm calling from Memphis, Colorado.
Thanks so much. The mention of Tetra Pak was a suggestion for a alternative to canned packaging, and the reason is there are some downsides to Tetra Paks, mainly in terms of recycling, that you can't recycle them, but they're made of multiple layers of materials, like cardboard and plastic and foil, but the inner lining is PET, which is the same material that makes up our disposable plastic water bottles, which actually looks to be relatively safe. It's hard to prove ultimate safety, but we've looked at contaminants. For example, I was involved in a study looking at chemicals that might leach from PET into drinking water, even stored at very high temperatures, and we saw relatively few contaminants. So in terms of just thinking about safety of the food contents, I think that's, in addition to glass, it's a reasonable alternative.
Oh, great. Thank you.
Thank you. We can probably take a couple of more questions or a couple of more comments. And when you are done asking your question, if you could press *2 to mute your line again, that would be appreciated. Go ahead.
Yes, I have a related question. Can you hear me?
Yes.
This is Chris Benson. I'm just an individual. I'm in Juneau, Alaska.
Welcome.
And I was curious about the canned foods. Is that just cans that are lined with a white lining, or all canned foods?
At this point, to my understanding, pretty much all cans are lined, and there's a good reason for it in a way, because it dramatically increases shelf life, because it helps seal the contents from air, and if the can is damaged, it's less likely to be contaminated, but it has this unfortunate downside. And I want to mention, too, there's actually some brands have gone to BPA-free can liners. At this point, I'm not convinced that any of the alternatives are great. We don't really know if they're safer, which is why at this point I try to find food that is not in cans, and that may change in the coming years. There are people working hard on that, but at this point I don't think there are very good replacements.
Okay. Do we have any other questions or comments?
Yes. Hello, can you hear me?
Yes.
Yes, hi, this is Dr. Luis Rivera with the federal government, ATSDR. I just had a quick question, very interesting talk. My main question relates to, there is definitely a plethora of chemicals out there that are sort of associated with development of breast cancer. My main question is, what would be sort of the way of concentrating the efforts into a specific, is there any hot sort of chemical of interest or emergent chemical that researchers and scientists should be focusing on, instead of just conducting many exploratory analysis to try to figure out all the possible connections?
It's a great question. I think where the people who are experts in this are looking, and I know we have some of those on the call who could chime in, is we're now understanding several of these mechanisms. For example, it's why I went into detail about the hormonal mechanisms of kind of tissue priming. So if we start to look for, and some groups are doing this, like NTP and the other collaborators working on TOX21 and TOXCAST, they're starting to screen chemicals for their activity at particular receptors, or their activity in ways that resemble other hormonally active compounds, in a way to sort of direct the search. So I think this area of tissue priming and developmental exposure is quite ripe for more investigation. And I'd invite experts on the call to weigh in if there's other specifics they want to talk about.
Great, thank you.
Thank you. I think we have time for probably one more comment or question, and then I do need to move on to some announcements to wrap up the call. So I'd like to invite anyone out there who would like to participate to go ahead and press *2 to unmute your line.
Hi there, it's Christina Hunter. I teach at the University of Manitoba here in Canada.
Hi.
Hi, I'm really interested and thank you so much for your talk. A couple of quick questions. First of all, I was curious about the lack of discussion around some pesticides and wondering if that was left out on purpose or you just didn't have time for it and so on.
Thank you. Yes, it's mainly a thing of time and also relative expertise, but there is a lot of investigation about, so some organic chlorine pesticides may be related. Atrazine, which is not a pesticide but is related, is of emerging concern for this developmental impact on breast development and the changes that can occur with that. So there is evidence that some pesticides may contribute to breast cancer risk, and there's a lot of interest in some other pesticides.
Yeah, okay, good. And then I was also curious about the nonstick cookware. My understanding was that the PFOA is being phased out.
Yes, but its relatives are not, and they don't appear to be much better.
Ah, I see. Okay, very useful. Thank you.
We call them, we now mainly refer to them as perfluoroalkyl substances, PFASs, because there are so many relatives, and people were thinking that the short carbon chains are better, but they're not so much appearing to be. They may break down faster and maybe metabolize more quickly, but it's not obvious that they're less harmful.
Wow. So is this really all nonstick cookware then that we should be concerned about?
Anything with a nonstick coating like Teflon.
Mm-hmm. Yeah.
I use cast iron and stainless.
There are some that say they're ceramic coatings, so I'm not sure about those.
Right. As I believe, I don't know for sure if those ceramic coatings are free of nonstick, but if they don't have a nonstick coating like Teflon,
mm-hmm.
I suspect they're free of perfluorinated substances.
Yeah, that's very handy. Thank you.
Thank you so much. You've given us so much to think about in our choices and what we can do. Meg, did you want to make any final comments before I move on to announcing the Alaska events?
I think I've said enough.
Okay.
Thank you.
Okay. So as I mentioned at the beginning of the call, we're thrilled that Dr. Schwarzman will be in Alaska next week, and her trip to Alaska is made possible with support from the Alaska Run for Women. Co-sponsors include YWCA Alaska, Planned Parenthood of the Great Northwest and the Hawaiian Islands, and the Alaska Nurses Association. The main community event is in Anchorage on Thursday, June 29th, 1 week from today, at the Lydia Selcraig Chalet at Russian Jack Park. So please join us for that. It's called Resisterhood, Stand Up for Our Health, and there will be a community reception at 6:30 p.m., and Dr. Schwarzman's talk and discussion will begin at 7 p.m. In addition, Dr. Schwarzman will be visiting Mat-Su Regional Medical Center and Providence Medical Center for grand rounds, and will be speaking at the Alaska Family Medicine Summit in Anchorage. And if you are a medical professional and would like more details about any of these events, you can call our office at 907-222-7714. And again, I want to thank all of you for calling in, and thank you, Meg, for your truly informative presentation. I'll probably be listening to it again for all of the things contained in the notes that you were reading about the details of what we can do. So thank you so much, and have a wonderful day, everyone.
My pleasure. Thank you.